All right, everybody, welcome
Happy
to the
New Year.
HIRM seminar. It's great to have everybody with us again, and a very warm welcome to our seminar speaker for today, Dr. Ling-Ling Chen. Dr. Chen received her Bachelor's of Science at Langzhou University in China, and then her Master's in Pharmacology at the Chinese Academy of Sciences in the Shanghai Institute, Materia Medica, and then moved to University of Connecticut Health Sciences Center, where she performed a PhD and a brief postdoc in Gordon Carmichael's lab. And then in 2011 became a principal investigator at the Chinese Academy of Sciences Center for Excellence in Molecular Sciences, and is currently associate director of the State Key Laboratory there. So it's amazing to have her join us all the way from China, and thank you for getting up very early in the morning there to present your seminar to us. We look forward to hearing about the role of processing, conformation, and function of long and circular non-coding RNAs. So welcome, Dr. Chen.
Thank you. Thank you, Frank and Jay, for this very kind invitation. Also, thank you all for switching the regular time at 3 p.m. to now to fit my time zone. I really appreciate that. So today we talk about how we link RNA processing, conformation, and function all together when we study long and circular non-coding RNAs. Okay, so now we know long non-coding RNAs are important regulators of gene expression networks. So in a cell, different long non-coding RNAs can actually regulate chromosome, territory, chromatin, transcription, post-transcription, translation, post-translation modification levels with distinct mode of actions. So just like proteins, for long non-coding RNAs to have function, they have to localize to specific subcellular localizations. And we have to link the RNA processing to RNA localization and function. So that being said, once the RNA is processed, they choose to their cellular fate. And then once they localize to where they act, they can form the right conformation where they can associate with the proteins to regulate to regulate cellular function. So today I will first share with you how we find the distinct RNA processing may lead to different functions of ncRNAs in the context of stem cell pluripotency. The conservation of ncRNA occurs at multiple levels. So this is a review article from Igor Volitsky, who had been trained with Dave Bartel a long time ago. I really like this review article from Igor. So where he proposed that the conservation of ncRNAs occurs at multiple levels, from the sequence of the ncRNAs to positionally conserved ncRNAs, and in a more broad definition, if the RNAs have the similar structure folding or have the similar functions, they also be considered as the conserved ncRNAs. But the question is whether processing of ncRNA autologs is conserved remains unclear. So my postgraduate student Chen Jieguo, she just had her PhD defense last November, and Xu Kanma, who is a computational people from my colleague Li Yang's lab, they work together to address this question. So we wanted to study the long non-coding conservation using human and mouse ear cells, H9 and R1. We choose nuclear and cytoplasmic localization of ncRNA autologs as outcomes of RNA process, as processing finally determine where they localize. So we get the RNAs from nuclear and cytoplasmic, and we did the RNA sequencing, looking for the different compartmentation of different ncRNAs. What we found is, as expected, mRNAs, those gray line or the dashed gray line, are localized in the cytosol as expected for translation. But for the long non-coding RNAs, we found in mouse cells, mouse ear cells, it's more preferred localized to the nucleoli. But in human ear cells, have more evenly distributed patterns. And we found the same is true for both sequence and positionally conserved ncRNAs. And this observation is independent of pluripotent states of ear cells. For example, we examine naive-like states or primed states of the ear cells. So the question is, are there any functional consequences of these distinctly localized ncRNA orthologs in the context of pluripotency? Because in mouse ear cells, these ncRNAs are localized in the nucleus. So we use CRISPR-Cas9 to knock out these long-non-coding RNAs. But we don't see any change or any detectable change of the expression of the marker genes OCT4 or non-OCT. However, in human ear cells, as these RNAs are localized in the cytosol, we just simply deplete these ncRNAs with shRNA. And we found this altered expression of pluripotent marker gene OCT4 or non-OCT. So this suggests that probably these distinct localized ncRNAs play different roles in ear cells. And some of these cytoplasmic localized ncRNAs, they probably play a role in the pluripotency. Then we focus on one of the abundant such ncRNA, which we named FAST. This RNA is derived from the antisense transcript of the FOXD3, this transcription factor. In human ear cells, FAST is fully processed to produce a mature long-non-coding RNA, which we call the human FAST, which is about 547 nucleotides long. In mouse ear cells, this FAST is the major isoform of mouse FAST is the unspliced isoform, which is largely localized in the nucleus. So we consider this ortholog pair as a conserved pair because it's conserved at both RNA sequence and positional transcription levels. So at the RNA level, human FAST contains about 230 nucleotide sequences to the unspliced major isoform of the mouse FAST. At the genomic level, up to 3 kb upstream of FOXD3, 74% of the genomic sequence is conserved. And these RNAs are positionally conserved because they both have the same transcription direction and conserved upstream and downstream sequences. And very interestingly, if we differentiate both human and mouse ear cells, we found a rapid decline of the FAST. Here it's just spontaneous differentiation, and in human ear cells, we did 3 different germ layer differentiation. And then we knocked down human FAST, shown here by Northern. Using shRNA, we can deplete the human FAST quite nicely. Here we found without the human FAST, human ear cells have this aberrant colony formation and also epistaining that is the indicator of pluripotency. We further confirm these phenotypes using CRISPR-Cas9 knockout. We also examined whether the cytoplasmic localized human FAST can encode a peptide or not, and we found human FAST does not encode a peptide, although it's localized in the cytosol. And we did the RNA sequencing. We found a loss of human FAST led to altered expression of pluripotent genes in H9 cells. As the most strikingly affect genes that are involved in the Wnt signaling pathway. And Wnt signaling we know is well established. In the absence of the Wnt, it forms a destruction complex where the anchor protein axin interacts with CK1, GSK3, and phosphorylated beta-catenin. It can also recruit the E3 ligase beta-trap, and beta-trap can ubiquitinize phosphorylated beta-catenin and degraded beta-catenin. In the Wnt-on situation, beta-trap is de-associated with this previously called destruction complex and free beta-catenin to be translocated to the nucleus, where they can activate the Wnt gene transcription. So we did the TSA human fast pull down assay. So TSA is a partner, allow us to do the affinity pull down, and then examine all the key factors in the Wnt signal pathway. And we found only the E3 ligase beta trap is associated with human fast. So when Yuhang, now Yuhang is a postdoc for about 2 years at MGH Hospital, he and Chen Jie together, they proposed that human fast may act as a molecular decoys that can interact with beta trap that prevents beta trap to be engaged with destruction complex. Whereas in the human fast knockdown situation, now is still the win-off condition to form the destruction complex. So to test this idea, Chen Jie ran this very elegant cytoplasm and nuclear fractionation assay, where she found that beta-catenin levels are significantly reduced in both compartments. And in the meantime, she also found the ubiquitination of beta trap is increased. And then they performed the pull-down assay with the anchor protein antibody axin-1, and they found that in the human knockdown, human fast knockdown situation, the increased beta trap with the axin. The question is, how does this non-coding work in human ES cells? So Chen Jie performed this in-cell shape map assay with the chemical probing NNI. So for the chemical probing, it's basically these chemicals can access to this bulge single-strand RNA region that does not really react with the regions have the double-strand RNA format or interact with the protein. So he found that human fast can form these 5 similar loop regions, and each loop using the in vitro binding assay can bind with beta trap. Okay, quite strikingly. Beta trap belongs to FBW protein family that contains 7 repeats of WD40 repeats at the C terminals. And she did the mutation of the beta trap protein. And quite strikingly, this interaction with beta trap requires all the 7 domains of the WD40. Showing here, this is the last, this is the minimal truncation she used. Without this 2 WD40 domain, now this mutant does not interact with human fast anymore. And this interaction is quite specific because there are quite several different WD40 repeats containing protein, like this one. This is a beta trap, and this one WDR26 that contains 6 different WD40 domain strap that have 7 different WD40 domains. And this human fast only specifically interact with beta trap, but not other WD40 domain containing proteins. Next question is human fast sufficient abundant to affect the levels of beta trap involved in Wnt signaling? So she did a quantification in H9 cells. There are about 3,000 beta trap in the cytosol. We calculate the human fast indeed contains about 140 copies. And if consider this, each ncRNA has 5 loops. Now there are about 700 copies of the ncRNA in H9 cells. So theoretically, more than 20% of cytoplasmic beta trap can be bound by human fast. Indeed, in different human cell lines, when we knock down human fast, now we see the decreased Wnt signaling target gene expression. So this suggests that human fast is sufficient to prevent beta trap to be engaged with destruction complex. Well, here shows the mouse fast. The major isoform of the mouse fast is the unspliced isoform. This full-length mouse fast does not interact with beta trap, okay, shown here, in vitro binding assays. And for this 230 conserved nucleotides, again, it does not really form this loop region. It's not conserved. So the structure of human and mouse is not, of this ncRNA is not conserved. So far, by looking at the subcellular localization of conserved ncRNAs, we found they are quite different. And we focus on one of the ncRNA human fast. We found the human fast ncRNA can promote Wnt signaling in human ESL propotency. So now the question is, what factors can regulate the distinct processing of ncRNA orthologs? And Chen did this ectopic expression assay. So basically, she cloned the full-length human fast and mouse fast by the promoter EF1 alpha, which can be expressed in both human and HNN cells. And we examined the expression levels of this construct. They're comparable. The quite amazing observation was human fast is processed and localized in the cytosol in HNN cells, but it's constant in the nucleus, although also expressed at a similar level. However, the mouse fast now is processed and located in the cytosol. But in R1 cell now, again, it looks like its endogenous format also retained in the R1 cells. So this suggests that not the cis elements, but some differential expressed trans factors can contribute to such distinct localization patterns. And then now it's a long journey for Chenjie. So we spent more than 1.5 years to figure out which trans factors may likely regulate this process. So the hypothesis would be either the enhancers or suppressors. So if we knock down the suppressors, we would see the decrease in nuclear cytoplasmic distribution of the RNA. But if we knock down the enhancers, we would see the increase in nuclear cytoplasmic distribution of the RNA. But first of all, we have to identify which factors that can potentially bind to the pre-RNAs of both isoforms. And in collaboration with Shanghai, we identified 70 common proteins might interact with both human and mouse fast, and then select 10 such RBPs that are differentially expressed in H9 or R1 cells. So when we do this here, we wanted to look for the inhibitors that are highly expressed in mouse ES cells, but wanted to find those enhancers that are highly expressed in H9 cells. And then Chenjie did the individual knockdown of each individual protein, and she found one of the factor called PPIE. Knockdown of PPIE appears to affect nuclear cytoplasmic localization of the fast. The PPIE, it turned out to be a quite new enzyme, but it was shown in the 2017 Nature paper that talks about spliceosome structure. And this protein is highly conserved. PPIE is an extremely abundant protein in R1 cells. We did the calculation again. There are 280,000 copies in R1 cells versus 80,000 copies in H9 cells. But indeed, if we look at the expression pattern by Western blot, we thought that probably R1 can even have more abundant PPI expression than that in H9 cells. Shown here by 3 different shRNA knockdown for PPIE, and here's the endogenous scramble treated H9 cell PPIE level. So compared to here, I think maybe more than 5-fold even. But nonetheless, PPIE knockdown resulted in the increased mouse fast export in all the 3 different shRNA treatment. That is accompanied with increased splicing of the mouse fast. Shown here, this is distribution of the spliced isoform nuclear and cytosol, this unspliced isoform upon PPIE knockdown. Now we see increased spliced isoform and decreased unspliced isoform in the nucleus. And PPIE seems not only responsible for human fast export. In our sequence analysis, we found PPIE knockdown also reduced the the distribution pattern of other positionally conserved ncRNAs in R1 cells, which can be largely rescued by reintroducing the PPIE into the PPIE knockdown cells, these gray bars. However, at the moment, we still don't know how exactly PPIE achieves its specificity on ncRNA processing. So now Chenji still focus on this question, and hopefully in the future, we can have some sense on understanding how PPIE can achieve this very specific ncRNA processing. So by looking at what factors can regulate this distinct processing of ncRNA orthodox in human and mouse ear cells, now we found PPIE is a new factor, which is splicing inhibitor, can regulate distinct fast processing in the context of propotency. And we also examined other ear cells in the primates, like the ear cell from rhesus monkey. We found both PPIE expression, fast processing, and its role in wind signaling is more similar to the human ear cells. So this altogether suggests the possibility that the conserved ncRNAs may achieve functional divergence through non-conserved arm processing. So I want to use this very interesting title. I really like the title written by Perel, conducting at the Rankin Institute, The Secret Life of the ncRNAs. They are conserved yet not conserved. This is also changing the way that we use animal models to study ncRNA function, because very often we come across that knockdown one ncRNA that have striking cellular function, but we don't see apparent phenotypes in the mouse models. So hopefully in the future, we can address more towards this direction. So the take-home message one is the conserved ncRNAs may achieve functional divergence through non-conserved ncRNA processing. However, we still have lots of functions remain to be addressed. For example, how is PPIE differential expression achieved in human and mouse ear cells? So how does PPIE can suppress RNA processing in different types of ES ES cells? And what other RBPs might be involved in this process? Now we are doing the screening. We found at least 7 different RMP factors can interact with PPIE, but we don't know whether they can interact with this protein simultaneously or at a different spatial and timing manner. And we also wanted to ask, now we don't see any role of mouse fast in the wind signaling, but does it have any function in mouse ear cells? And most importantly, we wanted to ask that similar ncRNA functional divergence exists in other contexts. So definitely future studies are needed to investigate more general ramifications of this phenotype in propotency and in other contexts. So here I talked about these ncRNAs, which were discovered from pervasive transcription in 2007 to 2010 by many people, John Ring, Harvard Chang, and Dave also did lots of pioneer works on this RNA's function and processing. But they were thought that most of these intergenic region transcribed ncRNA are polyadenylated except NEAT1 and MALAT1. So in 2008, Spectre Lab at Coastal Harbor found that the 3' ends of MALAT1 and NEAT1, they're actually the 2 very famous non-codons, very abundant and play important roles in splicing spicle and paraspicle function and assembly. And Spectre Lab found the 3' end of these 2 are not ended with poly A, but are rather processed by a tRNA-like RNP catalyzed mechanism. And later on, Stace and Willis labs have shown that their 3' ends are stabilized by triple helix structure. When I was student with Gordon, I luckily worked with NEAT1, and I found that NEAT1 is responsible for the paraspicle assembly. And this earlier observation by the Dr. Spector's lab indeed triggered me to ask the question, are there additional types of RNA species in the transcriptome? So we collaborated with Li Yang's lab. So he's a principal investigator at the Partner Institute of CAS and the Max Planck Institute at for computational biology in the same big campus in Shanghai. So we look at the non-polar identity RNA sequencing, aim to identify some previously unknown new RNA, new types of RNAs. So using this non-polar RNA sequencing, we found those intron-derived snow RNA capital non-coding RNAs and circular RNA derived from Larry introns, and also together with other people, later on we found the back splicing of the circular RNAs. And when we study the snow-link RNAs, we also found this polycitronic transcript-derived spar RNAs, which have the snow RNP at the 5' and 3' are polyadenylated. But overall, all of these are conventional processed link RNAs because compared to the intergenic region RNAs, they do not have their own promoters, but are processed from POL2 transcribed pre-RNAs and then stabilized by distinct mechanism, either by forming snow RNP complex at the end or by forming circular RNA structures. So I'm going to talk about the circular RNA here in the next 30 minutes. So the single-strand RNA can be in the circular formats were originally observed in 1976 in the plant virus. Here is the EM picture of the denatured viral RNA. It's a circle or like the tennis racket. And then group 1 intron self-splicing can also have the circular intermediate. In the beginning of 1990s, there are a handful of the circular were reported from the spliceosome. However, they were thought as aberrant splicing and don't have functional significance. Now, with the application of non-polar sequencing, and most with the RNA-treated RNA sequencing, led by Sharpless Norman and Nicholas Ravitsky, and we also did the RNA-enriched sequencing in 2013. We found this cyclotronic RNA derived from intranulates, but the widespread circular are actually from the backsplit circular RNAs. So circular RNAs, now we know they're broadly expressed in metasomes, but generally at low levels due to the inefficient backsplicing. They derive from middle exons of the gene. So in principle, the formation of the backsplit circular can somehow affect the linear RNA splicing. Now we know the biogenesis requires spliceosome machinery and can be modulated by both cis sequences and the transprotein factors. And once they are formed, they are largely localized in the cytoplasm. With people, so many labs now work on the circular RNAs. Now we found, and others found that circular RNAs, if they are localized in the nucleus, they have potential to regulate transcription. And they also can compete with the nucleotides to interfere with signaling pathways and spawn and scaffold micromolecules. And here shows a typical gene organization that contains multiple genes. In the kinetic splicing, the inclusion of all the exons forms the linear RNA. The back splicing catalyzes distal splicing sites. In the meantime, it can produce linear RNA with the circular RNA forming exons excluded. So in the beginning, we asked how does the spliceosome can recognize this special unfavorable reaction. And we and Jeremy's lab, we found that this the formation of the transient double R region cross-back splicing forming exons in the intron region can bring the distal splicing site to the close proximity that can facilitate back splicing. And then we did this nascent RNA sequencing. We found the splicing efficiency, the back splice efficiency is very low in cells, which is less than 1% of that of splicing. So this was reported in 2016. I remember that I was so disappointed at the time because it sounds like these cyclins are expressed at low levels. So what they can do in cells. But anyway, so we found, although require this the pairing of the intronic alu elements, but the regulation of CRNA formation indeed is regulated by computation of arm pairing. When the pairing forms in the individual intron, now it just promotes just regular processing of the splicing. But if the arm pairing occurs across flanking intron, now it's more likely induce the circular RNA. So shown here is the simple vector expression assay. Let's focus the vectors number 2 and number 3. So this full pair is the pair of the complementary sequences can significantly induce circular formation. By introducing the competition can dampen circular formation. So because of the wide repeat alus and competition of arm pairing, we found, for example, in human ES cells, more than 50% of the expressed gene loci, they can actually produce more than one circular arm, which we call attentional circularization. Shown here is a famous gene called DNMT3B, the DNA methyltransferases. Computationally, more than 10 circular can be produced from these markers, and 3 of them can be readily detected by northern blot. And also interestingly, because of alu elements are primate specific, we also found an interesting increased complexity of circular expression during species evolution. That said, we found, for example, in the different tissues of mouse and humans, normally human tissues have more circRNA than that in mouse tissues. But at the end, ultimately we want to ask, what do they do? But it's quite challenging to study their function because they are circular, because they have sequence overlap with linear RNAs. So challenging circRNA studies can occur at multiple levels, from annotation to validation to lots of gain function studies. So we ask a very fundamental, almost a naive question in cells. If they already produce the linear RNAs, why do they have to produce circular RNA that have the same sequences? So we decided to study the biogenesis. Just to tell you a little bit, we found the circular RNA biogenesis requires intronic cis complementary sequences, and they are also slowly produced. And when Xiang and Chuzhao in the lab, they begin to investigate what transfer factors can regulate circular RNA biogenesis, as well as stability and conformation. So Xiang set up this, now it's widely used vector, so where he inserted this reverse cherry with the iris in the reverted order, and this cherry is split by an EGFP flanking by a pair of complementary sequences. So this reverted cherry can be translated. Once become the circle, can be translated to the cherry, and EGLP can be translated, of course, to EGLP. So this is an efficient circular expression vector. We use this for genome-wide siRNA screening that allowed us to identify more than 100 RNA binding protein can potentially affect circular formation. Importantly, upon viral infection, we found circular biogenesis function can be regulated by the downstream RNA binding domain containing factor NF90, F110. So this protein bound to the intronic region normally promote circular formation, but upon viral infection, these proteins can rapidly exported to the cytoplasm where they engage with viral mRNA to inhibit viral mRNA translation. And in the meanwhile, we observed the global reduction of nascent RNAs upon viral infection. So this earlier observation somehow linked the circular biogenesis to nascent to the innate immunity, and also suggests that upon or during innate immunity, circular RNAs they are unfurled during this process. And later on, they found that circular, although very stable, but they can be rapidly degraded upon poly-IC simulation, and on poly-IC, not all the other simulators, and can trigger innate immunity. the half-life or the turnover of these examined sequences are very rapid, under 1 hour. And then we found the endonuclease RNA can rapidly and globally degrade the cyclin upon polylysis and virus such as EMCV treatment. And it turned out that circular RNAs, among 34 examined circRNAs, 76 of them exhibited this 16 to 26 base pair intraductional RNA region. Some of them contains only 1, while others can contain to 3 or up to 4 different intraductional RNA regions. And such structures are seen less frequently in their linear cognate RNA, and allow circRNA work as a group to bind and regulate the innate immune adaptation RNA receptor PKR, and indeed AF90, F110 as well. Because the story already published a while ago, I just want to briefly summarize what we found here. So we found this RNA that show you they can form the adaptation RNA region, interact, for example, here PKR. We think this as one way to keep the PKR from unnecessary activation. Upon cells are infected by virus, they can stimulate the dimerization of the endonuclease RNA and become activated. The active RNA can trigger degradation globally of circular RNAs, and then release the PKR to be engaged with viral adaptation RNA to form the dimer activation and trigger the downstream ERF to alpha phosphorylation inhibit protein synthesis. Excuse me. And meanwhile, they can also activate the interferon-related genes. So this is one example that we found circular RNA slowly produced, and they can form these interesting, unique structures and work as a group to interact with PKR, but then they can be rapidly degraded. So this feature allows the circRNAs as one class of RNA to regulate the immune homeostasis. And we also found the circular RNA PKR axis is altered in the patients with the autoimmune disease SLE. So here, shown here, the samples came from my colleague, Dr. Shanghai from Ranger Hospital. Here's normal PM, what's it called, PBMC cells, peripheral blood mononuclear cells. This normal patients and those SLE, we found augmented PKR phosphorylation, and from the sequencing samples, we also found global reduction of circRNAs in the 3 mononuclear types of cells, monocytes, B cell, T cells, isolated from SLE donors. Importantly, now when we overexpress this adaptation RNA region containing circRNAs, all its linear isoform, we found it can significantly reduce the PKR activation as well as its downstream EF to alpha phosphorylation. Not only PBMC cell, but also individual like T cells. We also found the downregulation of interferon and interferon-stimulated genes, and these genes are well-established markers for diagnosis of SLE. So this observation suggests an exciting potential of using circular RNA as a strategy for SLE. So can we use the exogenous circRNA to dampen adaptation RNA stimulate PKR activation? So this is a question being asked in the past almost 2 years. We have some progress, but not that stage yet. So interestingly, as meanwhile, have a Chang's group and Daniel Anderson's group at MIT, they use different ways to synthesize the RNA circles, and it appears that circRNA is synthesized by different approaches use different immunogenicity. So in Harvard group, they found exogenous circRNAs synthesized by T4 phage intron can potentially stimulate signaling via RIG-I, but M6 modification can discriminate self from non-self, therefore bypass cellular RNA sensors. In Daniel's lab, in the Anderson lab, and they found that RNA circularization by anabana catalytic intron can diminish immunogenicity. So the question is whether unmodified exogenous circRNAs can bypass circular RNAs, cellular RNA sensors. So Chuxiao, I was lucky because Chuxiao decided to stay in the lab to further chasing this interesting project. So Chuxiao, together with my student, young student Sikun, they tried many different ways. And here they were able to synthesize. So this is the linear RNA, the circularized RNA treated with RNA. They get beautiful circles, okay, in all the different methods. So now they're able to use the method to get the circular RNA without any immunogenicity, shown here interferon, TNF alpha, IL-6, RIG-I, triggered by poly-IC. Here we can see the stimulation, IVT products, yes. A linear RNA with 3' end phosphorylation, yes. But linear RNA alone, much more reduced stimulation. And here the circular we synthesized, and also other methods. We see the similar observations in HALA and 293 and A549 cells. And more importantly, and Truchel did this in vitro binding and stimulation assays, and compared to the existing PKR inhibitors to a P of C16, this in vitro synthesized circular without immunogenicity show more than 1,000-fold higher inhibitor effects on the PKR activation. It's quite dramatic. So it turned out that binding of the PKR to these RNAs can somehow really, alter the conformation of the circRNA. This is the in vitro ship map assay. So this is the RNA we work with. It has a 2 PKR regions over here. So this is the shape reactivities shown here. The blue region, no activity because they're already engaged with PKR and the upstream RNA region. And when we bind to PKR, we found this bulge now become less reacted with NI. When bound with this RNA with RIG-I, we don't see any change between the ion-associated region. In the other regions over here, these are also bulge regions. This green color shaded, we don't see much changes. So we figure out that for the circular RNA, because it's already double-stranded and bound to PKR, the binding with PKR won't see any change over here. But for this body that close to the double-stranded binding ring, now we see the changes. So this is yet another evidence to show that PKR really can bind to the circular RNAs. So now we're working on the effect of this immunogenicity-free circular RNA on the suppression of double-stranded RNA, stimulate PKR activation in animal models. And hopefully we can wrap up some interesting results soon. But my lab really work on cells. Once begin to work with animal models, we see how slow that is. But it's very interesting work with animals. Okay, so now overall for the circRNAs, now we know circRNAs can involve in the immunity regulation, but that works as group, right? So how to identify individual circRNA with functional potential? The challenges is methods are inadequate to discriminate circRNA from mRNAs. As we can imagine, if you use CRISPR-Cas9 knockout, that's inevitably going to affect the linear RNA. For this CDR1AS, this entire exon predominantly conform the circRNA, that's fine. A long time ago, we began to use this CRISPR-Cas9 technology to disrupt the intronic pairing. However, we cannot work with this for the large scale screening because it's really complicated and time cost consuming. People used to use RNA and shRNA to knock down the circRNA, but half of the RNA also targeting the linear RNA sometimes can cause off target. So for example, here, when postdoc Siqi Zhang's lab in 2017, she picked up 10 circRNAs and examined, 9 circRNAs and examined the effects using shRNA to knock down circRNA. The overall knockdown efficiency is about, by using the siRNA targeting back-expressing junction site, that is the only place that can discriminate circular from linear RNA. And she found the knockdown overall effect is about 35%. And it can also affect like 15% of linear RNA expression overall speaking. So with Feng Zhang and Patrick Su, they published the programmable RNA-guided RNA targeting type 6 CRISPR effectors. We noted that they said this system is intolerant to mismatches in the center city region that hybridizes with the targeted RNA. So we began to think about, can we adapt this system to knocking down ncRNA, because that probably can discriminate the background junction site better than shRNA. So we did the parallel experiments with two positional matched shRNA and BSG gRNA for each circle RNA. And quite dramatically, we found she first screened 8 different Cas13 protein, and she found RFX Cas13D was the best for ncRNA knockdown. And she found almost about 65% of ncRNAs are gone on average without too much affecting on the linear RNAs. And then we did lots of characterization of this system on targeting circular RNA. So the paper has been recently online published. So I'm gonna cut the long story short. So here shows one example that she did the tiling. It's clearly to see that when the BSG set in the central city region, that can effectively knock down circular RNA without affecting the linear RNA. The question is, why does this system works well for circular knockdown? So we think compared to linear, because in the meantime, we also try the linear, we don't see a similar effect. I mean, that effective effect on the linear knockdown by using the system. We think the overall conformation of circular is more stable and rigid than that of linear, as I just shown you over there. And once targeted by the GR and Cas13, such region structure in circular RNAs would continue to be targeted, and the linear RNA is different. And also, a long time ago, I showed you that backsplice efficiency is extremely low compared to linear RNA splicing. So once cleaved by the Cas13, the newly produced ncRNAs cannot compensate for the loss of ncRNAs, therefore leading to the consistent reduction of ncRNA by this system. So nonetheless, we begin to try to optimize this system to perform larger scale loss of function screens. Again, it's quite a task because it's different from the CRISPR gRNA for genomic RNA targeting. We again can only targeting this minimum region for the different screen to make the library. As I mentioned earlier, ncRNAs are expressed normally in the at the low levels. So we only focus on the top 10 highly expressed ncRNAs in multiple human cell lines, including 293, HeLa, and human ESL H9. Overall, we got over 700 ncRNAs in the GRN library in cell screening. And for each of the ncRNA, we designed 5 different gRNA with a paired control, which is each own paired control. And this over 700 ncRNA pool represent the most highly expressed ncRNA in examined human cell lines. I wanted to mention that this entire library was originally wanted to using human ESL's propotency again as a readout, and also wanted to use H9 differentiation to neuronal cells as readout because we know ncRNA are highly expressed in neurons. However, so far we are unable to make the RFX Cas13D human ESL lines. So this is echoes with recent publication that the RFX Cas13D probably somehow toxic for certain cell lines. Okay, but there's no problem for us to set up 293, HeLa, as well as HT29, and now we are doing many other human cell lines, so that's totally fine. Okay, so nonetheless, so now we basically so shows here, we wanted to first examine how long this system can be last in cells. We found ncRNA knockdown, 3 of them, ncFAM120A, ncPOLY2A, ncHIPK3. Knockdown of them can last to 30 days in the system. Okay, the linear RNA still remains okay. And for one of the RNA, we did the sequencing compared to shRNA and gRNA, and we found treating with this system allows quite specific. So basically, there's not too much compared to shRNA off-target effect. This also echoes the earlier publication from Zhang Feng's lab. And then we did the simple screening to look at the cell proliferation. So we have the stable expressed cell line, infected with lentivirus, and we did cell passage for day 13, and then we do deep sequencing to look for the cells still livable. And then we can also do in vivo screening by generating stable cell line, and then put the lentivirus infection to the cells, and 7 days later, we injected this pool of cells into nude mice, and then 22 days later, we did the gRNA library, and then look for the ncRNA in the gRNA library that have the altered expression. So cut the long story short, I'm not going to talk about this, how we did the computation calculation, because it's just published. I'll just show you some new features we found for ncRNA function. So first of all, we found the cell type specific effects of ncRNA on cell proliferation. So from this over 700 of ncRNAs, we only found some of them can act in 2 cell lines or more than 2 cell lines. And then we did lots of validation on this. Overall, we did almost 40 different ncRNA validation, and they do have cell-specific effect shown here. For each of the validation, we did 2 gRNAs. This shows the linear RNA. This shows their knockdown effects in the different cell lines. And we observe this cell-specific, for example, the purple color for HT29, knockdown of these ncRNAs, we see reduced cell proliferation, but not for other ncRNAs, not for other examined cell lines. And similarly, this is another 2 ncRNAs, knockdown of them, we see reduced proliferation in 293 cells, but not in HT29 or HeLa cells. And then we also found the common oncogenic ncRNA called ncFAM120A. In all the 3 examined cell lines, they always reduce cell proliferation, also showing the similar reduced proliferation in the nude mice. So mechanistically, this particular ncRNA localized to to the monoribosomes. So the control here is ncHIPK3, which is in the cytosol. And for the another translatable ncRNA, zinc 609 is largely enriched in the polysomes. And importantly, if we remove this ncRNA using gRNA, 2 of them, now we see its parental mRNA become more enriched in the monoribosome. Okay, so per cell, there are about 23 copies to 30 copies of these circular RNAs. And correspondingly over here, we see after shifting from this polyribosome to monoribosome, we see the reduced protein translation of its parental gene. And we found this circular RNA preferentially bound to the translational inhibitor IGF-2BP2. So we saw this binding and the localization of this ncRNA at the monoribosome can prevent the IGF-2BP2 binding to the parental mRNA, therefore promoting FAM120A translation. So why does this happen? We found that the FAM120A is M6A modified, and IGF-2BP2 is known can be recurrent to the modified RNA. So shown here, this is some in vitro assays with the increased M6 level of FAM120A. Now we see the increased binding with the IGF-2BP2. This is a competition assay between the between the M6 modified circular RNA. And the linear FAM120 with the same sequence. Apparently, with the increased M6 modification, it can outperform the binding between IGF2 with linear FAM120A. And we also wanted to examine whether there are any circRNA is important for the development. That's still the issue in the open question in the circRNA field. So in collaboration with my collaborator, Jinsong Li, the student Lin Zhang in his lab, just to inject the mRNA of RFF casetin D and get RNA at the zygote stage and allows the development towards the blastocyst. And then we selected 25 experimentally validated human circRNA in our screening, and these RNA and then look for the expressed circRNA in mouse, and we found this conserved 15 circRNAs. And we also looked for the single cell sequencing top 10 circRNA in mouse pre-implementation embryos that is also expressed in human. So overall, got 24 candidates circRNA. And for each of them, we did this procedure, and we found only one circRNA called circMan1A2. The loss of this circRNA can reduce the size of the cavity over here and can cause dysplasia of the development, the two of them. So we're very happy to see this RNA appears to be important for mouse embryo pre-implementation development. And at the moment, for this specific circRNA in the human ear cells, we see similar phenotype. So, so here's the take-home message too. So we identified, in addition to the ncRNA I talked about in the very beginning, by using non-polar sequencing, we found this very interesting unconventional processed RNA species, and focus on this backspliced circRNAs. So I show you there's infrequently spliced. They can regulate by both cis and trans factors, and their degradation and structure. Biogenesis can regulate PK activation in immunity, and the MACE regulation is related with autoimmune disease. And we're working on how and whether we can use circRNA to dampen PK activation. And finally, we recently developed the Cas13 system that can discriminate the backspliced circRNA from the cognate mRNA, which will likely facilitate functional study of circRNA in the future. So all in all, by studying circular long non-coding RNA, we definitely feel that our processing, subcellular localization, and conformation all work together indispensably, and they are important factors in determining their functions. So that's basically what we are doing in the lab, circRNA biogenesis. Another part of the lab working on the linker processing and function nucleokinesis. But all in all, we try to do our best to apply the advanced technology that allow us to understand our process and function, which in turn going to bring new biology and translation potential in biomedicine. But again, when we think about long non-coding function, we probably have to think about it, what kind of animals we are being, or models we've been using. Okay, it's pretty much what we're going to talk about today. I would like to thank Chen Jie, who did this beautiful work on our understanding how the ncRNA process in different contexts, and Xiang and Chu Xiao, they discovered circRNA in immunity, and Xiang and Siqi were together for the circRNA Cas13D screening, and collaborators Li Yang for computational biology, Nan for SLE patients, mouse biology with Jinsong and Yangming, and Ji, whatever, so many imaging acquisition facilities. Thanks all for the funding agencies. And also we are hiring for postdocs. And thank you and Happy New Year. Sorry, it takes longer time than I thought. That really don't talk about in English for a while. Apologize for this.
All right, that's fine. Thank you very much, Ling-Ling. That was great. There is some time for some questions. So please feel free to unmute yourselves and ask your questions, or type them into the chat and I can ask the question for you. So maybe I'll start while people are thinking. So you've had a problem, you say, making the human ES cells with the dead Cas9 13. But what about mouse ES cells? Can you make those?
Most ES cells, at least it's fine for if we do the injection, it's no problem. But
so it's not something about the lethality. It's not something toxic necessarily. It's more maybe cell-based.
Well, so first of all, in the very beginning, we saw it's probably the methods we use, like lentivirus, somehow can increase the toxicity to for the cathodic system in the human ES cells, because we know this is more fragile than other cell types. But we tried all different ways. We still cannot make it. We tried different cell lines, even the PEG system for introducing the system. But most ES cells we didn't try, but for the zygote, that's fine. We not only look at the cathodic system in long non-coding RNA or circRNA, we're also trying to knock down mRNAs. So it also works well for certain mRNAs that that can be targeted by the cathodic system. So far we don't know what happened, but in another experiment we're working with, put the cathodic system in just normal human cells, it can somehow form the phase-separated things. So it can form the accumulation over there. So I don't know whether it's particular Cas13 protein, the forming that thing make it more toxic, H9. I really have no idea, but just didn't work.
All right. There's a question from Priska Orby. She asks, what were the 3 different methods that you used to produce the circular RNAs in studying their different immunogenicity?
Yeah, I'm happy to talk about that, because this is actually the first time I didn't really list the methods, because there's a little bit of debate in the field. So I do not really want to offend the people at the moment. That's why I put the method 1, 2, 3. Maybe she can send me email at the moment. I would be happy to tell her or tell him. So, so sorry about that.
Yeah, so why don't you contact her offline? That would be great.
Yeah, please do so. Hi, Dave.
Any other questions? Go ahead.
Can I ask a question, Frank?
Yeah, sure, go ahead.
So I was trying to understand what you're saying about the stoichiometry of the human HFAST and its protein ligand. It sounded like you were saying that there's enough ncRNA there to bind about 22% of the protein. And were you saying that would be enough to cause an effect?
Okay, so first of all, beta trap, not all the beta trap are engaged in the Wnt signaling. So I believe beta trap can degrade other factors, but we don't know how much of the beta trap is in the destruction complex normally. So, but when we do the calculation, we can only do the calculation of the overall pool of the RNA and the protein. But as a readout, we found that knock down the human fast, we see not only the, this pathway, we examine the CD44 and other Wnt signaling targeted genes, we always see the reduction of the Wnt signaling. We also try the different stimulation that can be compensated by chemicals, GSK inhibitor, for example. So we believe human fast can really work by disrupting the Wnt signaling pathway, but I have no idea how exactly the beta trap engaged in the Wnt destruction complex can be sponged or decoded by human fast.
So you're saying that there's something about the localization,
yes,
that could help explain how few binding sites could have the effect. Is that
right? Yeah, that is the model. But beyond the model, as I said, we test. What is this here? So the panel F shown here, when knock down human fast, we always see the reduction of this famous Wnt target gene X2 CD44, the 2 different knockdown, different human cell lines. So it's there.
Right. So you have the biological effect, and then are you thinking that each of those loops is binding a different protein? A different molecule of protein?
I don't know how, well, at the moment, the model is one loop bind to one beta trap, but they can bind more beta trap. So in our recent study of slurred RNA with DDS21, we start to use single molecule turf assay to address how many RNA is really associated with a particular protein. EMSA cannot really tell you the outcome. So we did the single molecule turf assay. So one slurred RNA, for example, can interact. We found almost 15 DDS1 molecule. But we haven't done that thing with this fast and beta trap. You're absolutely right. The one loop will probably bind more than one beta trap. That's for sure.
Thank you.
Thank you for clarify this.
Can I ask a question?
Sure, go ahead.
So I'm interested in the interferon response and the innate immune. So I was wondering, I know that there's some data, I'm not like an expert in the field, that ALU sequences are enriched in double-stranded RNAs that bind to RNA sensors like RIG-I and MAVs. And I was wondering, first of all, do you think those tennis racket shapes are circular RNAs that haven't been completely backspliced that might have ALU double-stranded RNA that could be recognized by other innate sensors of double-stranded RNA?
I think the length of the upstream RNA matters. For example, RIG-I can be recognized by double-stranded single-strand RNA. MD5 absolutely long double-strand RNA, including those mitochondria encoded double-strand RNA. So ALU, most of the single ALU, well, so ALU itself not really from double-strand RNA region too much, but it had to be intramolecular long double-strand ALU. They form double-strand RNA region. But most of those ALU sequences I found a long time ago, one way to keep this ALU sequences to trigger in immunity to keep these RNAs in the nucleus, some of them in paraspicles. But now we know another thing is A2I editing. So edited ALU sequences probably can disrupt the double-strand RNA region to form the body region. So I think it's all connected, Jude. Double-strand RNA, A2I editing, the length of double-strand RNA. This is really a big question in the field.
Yeah, because mostly people who work on lupus would think that the receptors like the RIG-I might be more important than PKR or
actually this is
in the interferon response.
Sorry for the
I don't know.
First of all, for the circular RNA, so endogenous circular RNA like that, as I show you, one of the shape map assay, in vitro shape map assay with circular RNA incubated with the PKR or RIG-I, and we see only the binding with the PKR. Yeah, can alter that body region, but not RIG-I at all. And in our in vitro assay, we don't see any change of incubated circular RNA with RIG-I. For example, the Chi63 recognition as the activation of the RIG-I signal, we don't see that change. So circular RNA endogenously, I think, is more preferred to interact with the PKR pathway. But exogenously, I can tell you that we have data that synthesized by T4 phage intron or anaconda intron. They always introduce actual sequence, okay? This actual sequence lasts over 70 nucleotides or 140 nucleotides. So that's quite long. That probably one way can trigger the RIG-I.
Yeah, that would.
Yeah.
I think we're going to end it there since we're already 11 minutes past the hour. So let's thank our seminar speaker again. It was a really great talk. Thank you very much for presentation and, as I said earlier, for getting up early and Hope you have a great day. And to everybody else, join us again, next week for our future HIRM events. Great seeing everybody. Bye, Ling-Ling. Thank you very much.
Thank you. Thank you. Hey, Dave, may I talk with you for a second? Can I leave alone?
Yeah. Dave, I'm gonna. Oh, I think he's gone.
Oh, he's gone already? Well, that's fine.
Okay.
Yeah, because one of my students is applying postdoc in his lab. That's all right. I can
Sure.
Yeah, yeah.
All
That's
right.
all right. Yeah, thank you, Frank. Yeah.
Thank you. It was great. Bye-bye.
Thank you for hosting. Bye-bye.
Sure. Bye.